Hello, everyone. I'm just here to talk about a joint pilot project that we did at the Garvan and Genome One with AWS. So the purpose of the entire thing was basically just to see if we could build some in-house cloud expertise and customize our pipeline for use in the cloud. So the pipeline we would use at Garvan in the cloud, simple as that. The team that put this all together, the real powerhouse behind it was, of course, Aaron Statham, our Chief Bioinformatics Officer, and he's in charge of the pipeline generally that delivers results to patients. We also have Mark Panizzi, David Thomas, and Andrew Stone on the research side, getting the cohorts together and doing some great work on the actual research end, getting the funding, all of that. And of course, there's Binup Nanu and myself are the DevOps team for Genome One. So the two of us did a lot of work actually building the system with a lot of reliance on our excellent AWS team, Ben Thurgood and Jamie Nelson, who worked with us directly as partners, which was excellent and a great experience for all of us. So a very quick rundown of Genome One. Who are we? So this is the initial batch of XTen sequencing sites around the world. These are the early adopters of the Illumina XTen sequencing technology. And as you can see on the Southern Hemisphere, we're basically that lonely little dot on Australia. But the map today looks a bit more like this. So a lot more players have joined the team. A lot more people have bought into the Illumina solution. Nevertheless, Genome One remains the largest genome sequencing facility in the Southern Hemisphere. We've got a lot of great strengths in the research and science side of it. Some of the best researchers in Australia. And we have some great key strengths in software development and data mining, which forms a great deal of this genomics work. Thanks to the Illumina solution in-house, we can now sequence a genome in about a week. That's end-to-end the entire DNA strand of a person for about 1.5K Australian. So that leads me to the problem of scale. So Genome One, as you can see on the slide, has 12 high-throughput next-generation sequencers, which produce about 300 genomes per week, which we need to analyze. Every genome has to go through an analysis pipeline. And really, we can't compromise on the core values of that pipeline: accuracy, traceability, documentation, consistency, functionality, quality, obviously. These are patients. Their lives are impacted. We can't compromise there. But good science costs money, and we need to save money somewhere, which is where AWS comes in. And where we have to, of course, cut down our own internal processes, go lean, go agile with everything. We've built a lot of software development expertise in-house as a result to kind of build all of our own systems, put our own stamp on everything, and continually improve everything we do in-house. And up until recently, that hasn't included our infrastructure. And this project set out to change that. So software first, hardware last is, I guess the keynote gave me a good way of talking about it. Until now, we've kind of been building the plane while flying it. And software first, hardware last is kind of us changing that around a little bit. Now, hardware, as you all know, is a big investment up front, and it ages quite quickly. And we are no strangers to buying large amounts of hardware. So our bioinformatics pipeline used to all run in-house on our systems. And by treating hardware the way we do the rest of our code, by creating essentially infrastructure as code, we're able to actually change the entire model of how we view hardware, how we build it. So infrastructure as code means we define all of our infrastructure in code, we commit it to a GitHub repository, and we're able to actually build on that, continuously improving the infrastructure as well as the software, which leads to some great cost reductions, faster analysis speed, and reduces risk for us, because every time we build something, we spin up a whole new, fresh infrastructure for it. But what you're probably looking for is this last point, the 20 times cost reduction in compute that we observed as part of this project. And that really is a very key takeaway from this. So how did we get there? First of all, our bioinformatic processing pipeline, like I said, used to run in-house. We sequence whole human genomes, about 18,000 per year, and they all have to go through this pipeline. The genome produces about 80 gigabytes of raw data in FASTQ format when it comes out of the sequencer and so on. That data size just about doubles when it moves into the BAM format, and as it passes through all these applications. And finally, it comes out as a GVCF, which is a listing essentially of the variants between our genome, the one that we're looking at, and the gold standard. Now, I'm not going to go into the details because Dennis has done a really good job already explaining that. So if you didn't catch her talk, it'll be available on YouTube. But suffice to say that these genomes can each be treated as an individual unit, and they can all be processed independently without the processors talking to each other. So it's a great place to use parallel computing and massively parallelize the whole process to do them all at once. What we built to facilitate that is a Docker image. So if you run anything in-house, you know that your HPC typically will have the issue of compatibility. Everything you run has to be essentially built into that HPC architecture so that, well, essentially, you have to make sure that your code matches up with the infrastructure you plan to run on. Docker changes that. By adding a Docker container into the mix, that Docker container can contain everything you need to develop your software, to run it on any compute. And as such, we kind of containerized everything we needed, put it into this container. And what it does is really quite simple. Downloads the raw data of the genome from NCI, the supercomputing facility that is partnered with us, or from Amazon's S3. And then we run it through a sequence of applications, one after the other, which forms our bioinformatics pipeline, which we developed to the Broad Institute standard. And then finally, that goes back into Amazon S3 or NCI. It was optimized for a certain kind of instance, and I'll go into that. But suffice to say for now, that instance is a particularly beefy instance, and we use its capabilities to the max. The Docker container was shaped and configured to specifically match those optimized compute capabilities of that machine. And that machine costs us about $40 per genome on AWS. But by making our infrastructure as code essentially able to be spun up directly anytime we want, we can take advantage of Amazon's spot pricing. We can take excess compute from Amazon, run our Docker container there, and we observe a cost of about $7 to $10 per genome, even including the potential for losing the spot instance and having to run again. Now, this pipeline's first research application was in sarcoma. Of course, someone has to pay for it. Something has to be done with the pipeline for us to be able to test it. So what we tested it with was this study. Sarcoma, if you're not familiar with it, is a terrifying form of cancer, not least because it's hard to diagnose, in that sarcoma affects a lot of young Australians specifically, and those young Australians, we can't screen everybody for it, and we don't really know which part of the population to screen in order to determine who's at risk of sarcoma, which means that right now, mostly sarcoma is only found when it's too late to do much about it. This study seeks to change that by trying to find in the genome a sort of fingerprint for sarcoma. And here's how we're doing it. Essentially, we've taken two data sets, one which is 4,000 elderly, healthy people, and one which consists of 1,000 young adults who are affected by sarcoma. Now, as Denise said in her excellent talk and went into more detail about, essentially what we need to do in order to find a way of diagnosing sarcoma is to compare the two, right? So essentially what we're doing as part of the study, the MGRB dataset of elderly, healthy Australians is used to assess the genomic patterns associated with healthy old age. You can see this is what a genome of a healthy person looks like, and do data mining on that to kind of get a fingerprint in the computer. And you can provide that study as a sort of control for any other study that seeks to compare to those healthy genomes with an unhealthy genome, such as these ISKS genomes, which are young people affected by sarcoma. And so the point of the research study is to then take that information, compare the two, and see how that fingerprinting matches up. What's in the affected genomes, but also in the MGRB, the healthy elderly, is easy to be discounted. We can say this is unlikely to be causative, and everything that's not forms our fingerprint of what in a person's genome might predispose them to the condition. So our process in doing the actual study, step by step, looks a bit like this. We didn't set out to run this pipeline 24/7. We set out with the pipeline so as to basically take what we had internally, push it into the cloud in a way that we could replicate automatically rather than manually. We actually wanted to be able to throw this up and then get rid of it when we're not using it, and throw it up again with as many compute instances as we need anytime. And you'll find that we actually were quite successful at that. And really the way we did it was to start by first optimizing our Docker container to run very nicely on one particular sort of Amazon instance. Then we set up a queuing system. The queuing system had the job simply of funneling jobs through that pipeline. And then we set up a data mover architecture, machines to take data up and down when it's done, as well as when it's about to be processed. And then we set up a layer of monitoring to create visibility, accounting, and billing information. Now, at every step, rather than simply build on that, we took that and put it into Amazon's CloudFormation service. AWS CloudFormation allowed us to then make that replicable so that it looks a little bit like this. A bunch of JSON files. I should note you can also use YAML, but basically our infrastructure, rather than be servers in a box somewhere, looks like these files, simple code. I've cut it off here because the actual code isn't that important, but you can see that I've selected here a particular definition, right, the ECS cluster. What I can do in CloudFormation is simply to define every instance and every parameter of every Amazon piece of hardware or software that we use in our studies and turn it into just a code definition. But I can also create a definition of my own, like ECS cluster, and build another file that defines in detail how that ECS cluster runs, what kind of machines are in it, how many of them, what the rules are for growing the system, shrinking the system, and also how these behave. So when I start up an instance, what do I run on it? So you can see in the code here, I'm not going to be able to point to it. Oh, maybe I can. So you can basically see that we are specifying what each of these instances will run. So when an instance comes up, it will run that piece of script, and it's all nicely put together in CloudFormation for us, such that we can change exactly how the things behave, how many cores they have, and so forth on the fly. Now, how did we optimize to the instance? That part is simple enough. What you're looking at is a series of applications. So every colored bar represents one application in our research pipeline. Each of these applications has different memory needs, different compute needs, and each of them uses a certain amount of RAM or CPU, etc. And simply put, what we did in order to optimize to our compute instance was to tweak the parameters of our pipeline and our hardware such that we observed full capacity and full utilization of that instance with a good genome sequencing time overall. And on those less compute-intensive steps that we found full RAM utilization, but didn't end up going into virtual memory because a genome is very large and a lot of it should be processed in memory to get the fastest possible result. So what this gave us was essentially a pipeline perfectly optimized for that one type of instance. After that, we built our queuing system. We took Amazon ECS, which is the container service, And what we did was essentially upload our Docker image into ECS automatically. And you can do this as part of a Bamboo deployment. So we can actually change out the container that we use anytime. And we decided to run one container per instance. And that makes it very easy for us. ECS just spins up compute and hardware for us whenever we need to run a container. So we pick one container per genome, and that's just how it goes. We also create an auto-scaling group, which essentially just means as the queue fills up, the compute cluster scales up to allow us to do it all in parallel. Now, we have an Amazon RDS database, and the point of the Amazon RDS database is simply to assign one genome to one task in the database and to keep track of the jobs that are running. So we can say if there is a genome inside the RDS database and it's not been sequenced yet, then we can go one step further and say this genome is now a running job. So we have the Amazon SQS service simply watches that RDS, looks for genomes that haven't been processed and aren't running, pulls them out of the RDS, assigns that genome to an instance, auto-scales up the Amazon ECS in order to pull down that genome and start processing it. That becomes a job in the RDS, and then the RDS takes it, the RDS watches, the ECS takes it from there. If the job fails, it'll go back into the queue. If it passes, it gets ticked off in the RDS until there's no more genomes left to be sequenced. So this is what the architecture looks like. Now, on the left, you can see NCI. That's really just a data point. It's where we stage our genomes. And then Garvin on the right is where we do our development on our containers, and we push them into the cloud system. In the center, you see one big Amazon VPC. And basically, what you can see on the right is just our continuous improvement architecture. We have all of our containers moving into Garvin's Bamboo system. Now, Garvin's Bamboo system is basically just keeping versions of all of our containers and allowing us to deploy a particular version at any given time, which is helpful if you need to, because we keep track of the exact pipeline that was used for each patient, and we need to be able to go back and replicate that. So this pipeline results in, essentially, from the right, the versions of our Docker container pouring into Amazon's ECR, their container registry, and then that container registry puts them into ECS, which is from where they go into the main loop of this. And the main loop is simple enough. You can see on the left the genomes coming in through the internet gateway. We have a set of NAT machines, whose entire job is to pull data down and then push it into the process. And then you have a private VPC, or rather a private subnet within the VPC, which is an auto-scaling group, runs our container at scale. And you can see the RDS is in that same bundle. And then there's a simple queuing service on the bottom right that acts how I've just described. So once they're all done, any given instance will push its data into an Amazon S3 bucket through that S3 endpoint at the end there. And then from there, the S3 bucket simply fills up over time. And once it's filled up sufficiently, we can pull it down over Direct Connect or put it on an AWS Snowball and push it to Garvin directly. So to keep track of this, we have used a set of information radiators that we basically built ourselves. So on the left, you can see the colorful wallboard that we used, and that's really just a very simple HTML setup with squares using Amazon's REST interface to query for the amount of spot instances we're using, the pricing that we've observed on each spot instance, how many jobs are running, how many are done. All of those simple metrics are viewable at any given time on a big wallboard in the middle of Garvin, and we never lose sight of those. as a side note, we used to handle this with a simple chicken interface, which you can see on the bottom right. The chicken was simply squeezed every time a genome went through, and the entire team would be alerted. We've just sequenced the genome. We've gone well beyond the chicken since then, but the actual process is the same. We're going into the statistics in a readable and human-friendly way. And at the top there, you can see a little tiny snippet from our Bamboo logs. You can see Bamboo's keeping track of every build of our Docker container. You can see that Bamboo is also tracking whenever we push code, what versions we're running, what versions we've put the patients through, and so on. So we can keep things consistent, auditable, and clear. And finally, on the right, you have the billing. And we can keep very nice track of billing because we don't have to tag everything manually. Since all the infrastructure is built by CloudFormation, we can set CloudFormation to simply tag everything for us with the appropriate billing tags so that when it comes time for someone to pay for it, we know exactly who used what compute and what data came from whom, and so on. On top of that, our pipeline has now run about 4,500 whole genomes, which is pretty impressive. That raw data is currently staged on NCI, and you can already go online to sgc.garvan.org.au to see that data for yourself. It's all browsable publicly, obviously de-identified, but it's all available to you and all Australian researchers. Data has been processed on spot, and we've been using a max spot bid of about $2.9. And I'll go into the details of the costing for the first phase of this over the next couple of slides. Now, after the project is finished, the results are pulled back down to NCI, and we have the following egress options. The direct connect from AWS to NCI is already set up. So if you're using NCI alongside AWS, the two can talk directly, and you get a much better egress cost coming down. Direct connect between AWS and Garvan flows through UNSW. So we can also pull our own data into Garvan at a fraction of the cost. And finally, the AWS Snowball we found has about the same costing as the Direct Connect does. So we can have the data physically shipped to us for about the same price. Finally, there's an egress waiver available for research data like this, which was covered in the keynote. So if you're a research institution, you too have access to improved pricing when it comes to pulling your data back down. Now, our phase 2 metrics, so for the most recent phase of the pipeline, are that we put through a staggering 2,859 high-quality genomes, roughly 70 million loci genotyped in each, and we found that there was about 3 million CPU hours used to process about 1.1 petabytes of data. So the pipeline is working beautifully at scale, and I can show you here for phase 1, for the first 856 of those genomes, how the actual spot price went for us. So we found that every genome went through in about 22 hours. The spot instances took a maximum of 4 tries to run the thing if you lost the spot instance, for instance, as you were going. But we found that a lot of our losses in terms of losing instances came from out-of-memory errors because we'd under-provisioned the RAM in some cases, and also from accidental terminations. So beautifully, it only took us about 1,040 jobs and attempts to get through the entire data set at a much better price. So this is the fluctuation we observed in spot price. We found that the pricing stayed constant if we pushed it even as high as about 150 C3-8X large instances and 200 R3-8X large, which are huge. That's 10,000 cores we can utilize before Amazon even starts charging us more. So as long as you keep a maximum spot price, you'll find that there's no problem getting the exact same costs that we did. One thing that we observed cost us a bit more than we expected was the NAT instances. And all I can say to that is the larger instances have much bigger bandwidth, but of course they'll cost you more. And we've worked with our AWS team since, and they've suggested a few nice fixes, along with the possibility of using Squid proxy. Of course, if you go straight across Amazon, you don't need the data movers at all. But since we used the data movers, that came to about 2.4K out of our total budget. You'll find that for 856 samples, the first 856, we spent about $15,000 on compute. It's about $17.6 per genome. Our data egress costs came to about $8.7 per genome, and then stashing everything on S3 gave us an extra 2.2. So that's in US dollars, as Amazon reports it. So that comes to about 37.33 Australian dollars per genome, which is quite a great price from our perspective. So that's basically it. That's our pipeline. And you'll find that it's basically already optimized for the cloud. It's already doing everything it's supposed to, but there's still more that we can do with it. We're able to actually adjust our data metrics and also have the possibility of using more instance types. And that one is something I really want to talk to. I mean, first of all, the data metrics were a mistake on our part, caused a lot of out-of-memory errors, which ended certain jobs before their time. We'll find much better results having fixed that problem. But using more instance types allows us to use Spot to the fullest. If we develop more Docker containers, we're able to use those Docker containers, optimize them for different instance types, and have AWS Spot Fleet pick out the particular types of instances that would be best for us, and just assign the appropriate container to each, which could save us even more money. Finally, phase 4, we could optimize out the NAT instances, use Squid proxy, or get rid of them altogether. And finally, there's an exciting new option available called AWS Batch. So I'll have Ben come up and speak to that. But first, I'd just like to give a quick special thanks, shout-out to our sponsors. So New South Wales OHMR gave us a great deal of funding for this research study. Asprey provided us samples, and so did the 45 and up study. Oops. And of course, I'd like to thank Amazon for giving us special pricing as a research entity, and for the invaluable assistance of Ben and Jamie. Over to Ben.
Thanks, Liviu. Okay, almost there. Well, I'm going to give you an introduction very quickly to Batch, the AWS Batch, which is a new solution from Amazon for batch processing. Very quickly in the last few minutes. So first of all, can I get an indication from anyone in the room who's actually put together some kind of batch workload themselves before? Yeah, quite a few of you. And you would know, as Liviu kind of alluded to, it's been a real pleasure working with Genome One and the Garvan Institute, because they kind of make really complex things look easy. But these things are actually still quite complicated, even though they're well understood and we've done it so many times. And AWS themselves have built so many different batch computing engines to create the services that we use. You know, there's using Spot Fleet and SNS and SQS, S3, DynamoDB, and creating all of the automations around this stuff is something you'll have to create and then also maintain. So AWS has released AWS Batch, which is a managed service for taking all of that undifferentiated heavy lifting off your hands and making it much simpler to do. It will handle installing all of the server, setting up all of the servers, managing the compute, managing the cost optimization for you, managing Spot Market, and also integrating in with lots of AWS services like S3, DynamoDB, and even Recognition to create quite complex pipelines, all with a managed service. So just a few concepts to go over. With AWS Batch, you create jobs. So you create a job definition that defines what vCPUs it's going to use, what image it needs to use, what storage it's going to mount, and other sort of characteristics of the job. And then when you submit a job to a job queue, it'll be automatically taken up by the scheduler and run. in the appropriate compute environment. So this is an example architecture. It's pretty common. We'll see input files being put into S3, triggering a Lambda function that calls submit to creating the job in batch, which puts it on a runnable queue. And the scheduler in batch will take up those jobs and put them into compute environments that match those computing needs. The results being put into S3, where they can then be used for analysis and those types of things. Now, one of the great chances for optimization in Genome One at the Garvan Institute and the genomics pipeline that Livia was talking about is taking these, each application, as he said, has specific compute requirements. So one needs lots of CPU, one needs lots of memory, one needs lots of I/O. And by breaking those down into individual jobs, we can use AWS Batch to schedule them into compute environments that match the computing needs of each of those jobs. So there's going to be some great opportunities for improvements into the future. So just finishing up there, that the AWS Batch is going to be able to give you a managed service to manage your batch workloads that can be cost optimized, that can either be resource optimized or can be time optimized if you've got a deadline, and it'll take away a lot of that work that you'd otherwise need to do. Thank you very much.